A new dawn in treating devastating brain diseases. In the realm of medical research, few breakthroughs capture the imagination quite like the prospect of replacing damaged brain cells with healthy ones. For decades, this concept has remained largely in the realm of science fiction, hampered by the brain's natural defenses and the complexity of neural tissue. However, recent developments at Stanford Medicine have brought this vision closer to reality, offering unprecedented hope for families facing some of the most devastating genetic diseases known to medicine. The human brain, with its intricate network of billions of neurons and supporting cells, has long been considered one of medicine's final frontiers. Unlike other organs that can be transplanted or tissues that regenerate naturally, the brain presents unique challenges that have frustrated researchers and clinicians for generations. Its protective barriers, immune surveillance systems, and delicate cellular architecture create an environment where traditional transplantation approaches often fail spectacularly. Yet within this fortress-like organ lies a population of cells that may hold the key to treating numerous neurological conditions. These cells, called microglia, serve as the brain's resident immune system and maintenance crew. They patrol neural tissue, cleaning up cellular debris, fighting infections, and maintaining the delicate balance necessary for proper brain function. When these cellular custodians become diseased or dysfunctional, the consequences can be catastrophic, leading to progressive neurodegeneration and ultimately death. Understanding the enemy, lysosomal storage disorders. To appreciate the significance of recent advances in brain cell replacement therapy, one must first understand the devastating diseases it aims to treat. Tay-Sachs and Sandhoff diseases belong to a class of inherited disorders known as lysosomal storage diseases, conditions that strike at the very heart of cellular function. These disorders affect the lysosomes, microscopic organelles within cells that function as recycling centers, breaking down worn-out proteins, carbohydrates, and lipids into reusable components. Imagine a city where the garbage collection system suddenly stops working. Initially, the accumulation might be barely noticeable, but over time, waste would pile up until normal functioning becomes impossible. This analogy captures the essence of what happens in lysosomal storage disorders. When genetic mutations prevent lysosomes from producing essential enzymes, cellular waste accumulates to toxic levels, eventually killing the cells and causing organ dysfunction. In Tay-Sachs disease, which predominantly affects children of Ashkenazi Jewish descent with a frequency of approximately 1 in every 3,700 births, the missing enzyme is called hexosaminidase. A Sandhoff disease, though less common, involves deficiencies in similar enzymes. Both conditions primarily affect the nervous system, where the accumulation of undegraded substances leads to progressive neuronal death and devastating neurological symptoms. The cruel nature of these diseases lies in their timing and progression. Affected infants typically appear normal at birth and may develop normally for the first few months of life. Parents celebrate milestones like first smiles, attempts to roll over, and early vocalizations, unaware that their child carries genes that will soon steal away these achievements. As cellular waste accumulates beyond critical thresholds, neurological function begins to deteriorate rapidly. Children lose the ability to see, hear, move, and eventually breathe independently. Most die within the first few years of life, leaving families devastated and medicine frustrated by its inability to intervene effectively. The microglia mystery. One of the most puzzling aspects of lysosomal storage disorders involves the relationship between different cell types in the brain. While neurons bear the brunt of clinical symptoms, suffering degeneration that leads to blindness, paralysis, and cognitive decline, they are not necessarily the most severely affected cells at the molecular level. This paradox has intrigued researchers for years and has led to important insights about how these diseases progress. Microglia, the brain's immune cells, actually contain enzyme levels that can be 1,000 times higher than those found in neurons. This discovery initially seemed counterintuitive. If microglia have such high concentrations of the enzymes that are deficient in these diseases, why do neurons suffer the most dramatic consequences? The answer lies in understanding the specialized roles these different cell types play within the brain's ecosystem. Microglia function as the brain's professional cleaning crew, constantly surveying their environment for signs of damage, infection, or cellular debris. This role requires them to maintain high levels of degradative enzymes because they must efficiently process large quantities of cellular waste material. They engulf dead cells, clear protein aggregates, and eliminate pathogens that might threaten brain function. In essence, they are the brain's waste management specialists, and like any intensive recycling operation, they require powerful tools to do their job effectively. Neurons, in contrast, are primarily focused on information processing and transmission. While they also need functional lysosomes for basic cellular housekeeping, their enzyme requirements are typically much lower than those of microglia. However, when lysosomal storage disorders strike, both cell types suffer, but in different ways. Microglia lose their ability to perform their cleaning functions effectively, while neurons accumulate toxic waste products that interfere with their ability to transmit signals and maintain their structural integrity. This understanding led researchers to hypothesize that restoring function to microglia might have therapeutic benefits that extend beyond these cells themselves. If healthy microglia could be introduced into diseased brains, they might not only resume their cleaning duties but also somehow support the health of surrounding neurons. Previous attempts and their limitations. The concept of replacing diseased immune cells with healthy ones is not entirely new to medicine. Hematopoietic stem cell transplantation, commonly known as bone marrow transplantation, has been used for decades to treat various blood disorders and some inherited diseases. The procedure involves eliminating a patient's diseased immune system and replacing it with healthy donor cells that can produce the missing proteins or enzymes. For lysosomal storage disorders affecting the brain, early attempts at this approach involved systemic transplantation with the hope that some of the new immune cells would migrate to the brain and establish themselves as healthy microglia. The theoretical basis for this approach was sound. If healthy precursor cells could reach the brain and differentiate into functional microglia, they could potentially restore the missing enzymatic activity and halt disease progression. However, reality proved far more challenging than theory. The brain maintains strict control over which cells can enter and establish residence within its borders. The blood-brain barrier, a sophisticated security system that protects neural tissue from potentially harmful substances and pathogens, also excludes many therapeutic cells that might otherwise provide benefit. Even when donor cells manage to reach the brain, they often struggle to establish themselves in sufficient numbers to make a meaningful therapeutic difference. Additionally, systemic transplantation approaches carried significant risks and limitations. The process of eliminating a patient's existing immune system, known as preconditioning, involves powerful chemotherapy drugs or radiation treatments that can cause severe side effects and leave patients vulnerable to life-threatening infections. Furthermore, the use of non-matched donor cells often triggered either rejection of the transplanted cells or graft versus host disease, a condition where the donated immune cells attack the recipient's healthy tissues. These challenges meant that even when partial success was achieved, the benefits often came at an unacceptable cost in terms of patient suffering and treatment-related complications. For children already facing devastating neurological diseases, subjecting them to additional trauma from toxic preconditioning regimens raised serious ethical questions about the risk-benefit ratio of such interventions. A targeted breakthrough. The revolutionary approach developed by researchers at Stanford Medicine represents a fundamental shift in strategy from previous transplantation attempts. Rather than trying to replace the entire immune system and hoping some cells would find their way to the brain, the new technique focuses specifically on the brain itself, creating a targeted therapeutic intervention that minimizes systemic toxicity while maximizing local effectiveness. The breakthrough involves a carefully orchestrated sequence of steps designed to create optimal conditions for donor cell engraftment while avoiding the major complications that have plagued previous approaches. The process begins with targeted depletion of existing microglia using specialized drugs that specifically eliminate these cells without affecting other brain cell types. This creates vacant niches that incoming donor cells can occupy, much like clearing space in a crowded parking lot to ensure new vehicles have places to park. Simultaneously, researchers apply focused brain irradiation to further prepare the tissue for incoming cells. Unlike the whole-body radiation used in traditional bone marrow transplantation, this targeted approach affects only the brain region, minimizing damage to other organs and reducing overall toxicity. The irradiation serves multiple purposes. It helps eliminate any remaining diseased microglia, creates additional space for donor cells, and may also modify the local environment in ways that promote engraftment. The next crucial step involves the direct injection of specialized donor cells called microglia precursors into the brain tissue. These cells represent a more mature subset of hematopoietic stem cells that are already committed to becoming microglia, making them more likely to successfully establish themselves in their new environment. Importantly, these donor cells do not need to be genetically matched to the recipient, which dramatically expands the potential donor pool and eliminates the need for time-consuming genetic matching procedures. Perhaps most importantly, the technique incorporates the administration of immunosuppressive drugs specifically chosen to prevent rejection of the donor cells without causing widespread immune suppression. These medications block the activation of immune cells from other parts of the body that would normally attack the foreign donor cells, creating a protective environment that allows the transplanted microglia precursors to establish themselves and begin functioning. Remarkable results in animal models. The results achieved using this targeted approach have exceeded even the researchers' optimistic expectations. In laboratory mice engineered to develop a version of Sandhoff disease that closely mimics the human condition, the brain-specific transplantation technique achieved remarkable success in multiple measures of therapeutic efficacy. Most impressively, the procedure resulted in the successful engraftment of donor-derived microglia throughout the brain, with more than half of all microglial cells being replaced by healthy donor cells. This level of engraftment far exceeds what has been achieved with previous approaches and represents a threshold that appears necessary for meaningful therapeutic benefit. Even more encouraging, this engraftment proved to be stable and long-lasting, with more than 85% of microglial cells still derived from donor sources 8 months after the initial transplantation. The therapeutic benefits observed in treated animals were dramatic and sustained. While untreated mice with Sandhoff disease typically died within 135 to 155 days, many animals receiving the brain-specific microglia transplantation lived well beyond 250 days when experiments were concluded. This represents nearly a doubling of lifespan, a level of benefit rarely seen in experimental treatments for neurodegenerative diseases. Perhaps even more significant than the extension of lifespan were the improvements in quality of life and neurological function observed in treated animals. Mice that received the therapy maintained normal exploratory behaviors, showing curiosity about their environment and engaging in typical social interactions with other mice. Their muscle strength and coordination remained substantially better than untreated animals, allowing them to move freely and engage in normal mouse activities for extended periods. These behavioral improvements suggest that the therapy is not merely prolonging survival but actually preserving neurological function in meaningful ways. For families facing lysosomal storage disorders, the prospect of treatments that could maintain a child's ability to interact with loved ones, explore their environment, and experience some quality of life represents hope beyond mere survival statistics. Unexpected mechanisms of action. One of the most intriguing discoveries from this research involves the mechanism by which transplanted microglia appear to benefit surrounding neurons. When researchers examined the brains of treated animals, they found that the lysosomal enzyme produced by the healthy donor microglia was not confined to these cells alone but could also be detected within nearby neurons. This finding suggests that the therapeutic benefit extends beyond simply restoring microglial function to include a form of cross-correction where healthy microglia somehow share their enzymatic activity with neighboring neural cells. While the exact mechanism remains to be fully elucidated, researchers suspect that microglia may be packaging the essential enzyme and secreting it into the extracellular space, where it can be taken up by neurons that desperately need this molecular machinery. This cross-correction phenomenon has profound implications for understanding both the normal physiology of brain cell interactions and the potential therapeutic applications of microglia replacement therapy. It suggests that microglia may play previously unrecognized roles in supporting neuronal health, serving not just as immune cells and cleanup crews but also as suppliers of essential cellular components to other brain cells. If this mechanism proves to be as robust and widespread as initial evidence suggests, it could explain why relatively modest levels of microglial replacement can produce such dramatic therapeutic benefits. Rather than needing to replace every diseased cell in the brain, it may be sufficient to restore function to the microglia, which can then support the health of surrounding neurons through this enzyme-sharing mechanism. Implications for human translation. The potential for translating this breakthrough from laboratory animals to human patients appears promising, largely because each component of the therapeutic approach builds upon medical techniques that are already in clinical use for other conditions. Brain irradiation is routinely employed in cancer treatment, drugs for depleting specific cell populations are used in various therapeutic contexts, and immunosuppressive medications are standard components of organ transplantation protocols. This familiarity with the individual components of the treatment regimen means that researchers have extensive knowledge about dosing, timing, and potential side effects, which should accelerate the development of human clinical trials. Rather than having to develop entirely new therapeutic approaches from scratch, the Stanford technique represents a novel combination of established medical interventions, each of which has been refined through decades of clinical experience. The fact that the approach does not require genetically matched donor cells represents another significant advantage for clinical translation. Traditional bone marrow transplantation often involves lengthy searches for compatible donors, during which patients may deteriorate significantly or become too ill to tolerate the procedure. And off-the-shelf therapy using readily available donor cells could dramatically reduce the time between diagnosis and treatment, potentially intervening before irreversible neurological damage occurs. Furthermore, the brain-specific nature of the intervention means that patients would avoid many of the systemic complications associated with whole-body immune system replacement. This could make the therapy suitable for a broader range of patients, including those who might not be healthy enough to tolerate traditional bone marrow transplantation procedures. Broader applications in neurodegeneration. While the immediate focus of this research involves rare lysosomal storage disorders affecting children, the implications may extend far beyond these specific conditions. Many common neurodegenerative diseases share fundamental mechanisms with lysosomal storage disorders, including the accumulation of toxic cellular waste products and the dysfunction of brain immune cells. Alzheimer's disease, for example, involves the accumulation of abnormal protein aggregates in the brain, and microglia play crucial roles in attempting to clear these toxic deposits. As the disease progresses, microglia become increasingly dysfunctional, losing their ability to effectively remove harmful proteins and even contributing to neuroinflammation that accelerates neuronal death. Similar patterns of microglial dysfunction are observed in Parkinson's disease, Huntington's disease, and numerous other neurodegenerative conditions. This suggests that microglia replacement therapy could potentially benefit millions of people suffering from common age-related neurodegenerative diseases, not just the relatively small number of children born with lysosomal storage disorders. If healthy microglia could be introduced into the brains of patients with Alzheimer's or Parkinson's disease, they might restore the brain's ability to clear toxic proteins and reduce harmful inflammation. The concept is particularly appealing because it addresses fundamental mechanisms of neurodegeneration rather than merely treating symptoms. Current therapies for most neurodegenerative diseases focus on managing symptoms or slightly slowing progression, but they do not restore lost cellular functions or reverse underlying pathological processes. Microglia replacement therapy offers the tantalizing possibility of actually repairing some of the cellular dysfunction that drives these devastating conditions. Challenges and future directions. Despite the remarkable promise shown by this breakthrough approach, significant challenges remain before microglia replacement therapy can become a clinical reality for human patients. The complexity of human brain anatomy and physiology may present obstacles that are not apparent in laboratory mouse models, and scaling up the procedure to accommodate the much larger human brain will require careful optimization of injection techniques and cell dosing protocols. Safety considerations will be paramount in early human trials, particularly given that the therapy involves direct intervention in the brain of patients who may already be neurologically fragile. Researchers will need to establish optimal treatment protocols that maximize therapeutic benefit while minimizing the risk of complications such as bleeding, infection, or unintended effects on normal brain function. The timing of intervention represents another critical consideration. In progressive neurodegenerative diseases, there may be a therapeutic window during which microglia replacement can provide meaningful benefit, but after which too much irreversible damage has occurred for cellular replacement to be effective. Identifying this window and developing methods for early detection and intervention will be crucial for maximizing the therapy's potential impact. Additionally, researchers must continue investigating the long-term consequences of microglia replacement, including whether the transplanted cells maintain their function over time and whether repeated treatments might be necessary to sustain therapeutic benefits. Understanding these factors will be essential for developing comprehensive treatment protocols that can provide lasting improvements in patient outcomes. A new chapter in neuromedicine. The development of targeted brain cell replacement therapy represents a watershed moment in the treatment of neurodegenerative diseases. For the first time, researchers have demonstrated a practical approach for replacing diseased brain cells with healthy ones while avoiding the major complications that have historically made such interventions impractical or impossibly risky. The success of this approach in animal models of lysosomal storage disorders provides concrete evidence that the brain's supposedly impenetrable defenses can be navigated safely and effectively when the right combination of techniques is employed. More importantly, the dramatic therapeutic benefits observed in treated animals demonstrate that cellular replacement can produce meaningful improvements in both survival and quality of life. As research progresses toward human clinical trials, the scientific community watches with cautious optimism. The families of children facing devastating genetic brain diseases have reason for hope, while the millions of people affected by common neurodegenerative conditions may eventually benefit from therapies built upon the foundation established by this groundbreaking work. The journey from laboratory breakthrough to bedside therapy is never simple or guaranteed, but the Stanford research has provided a roadmap for translating the promise of brain cell replacement into clinical reality. In doing so, it has opened a new chapter in neuromedicine, one in which the brain's most devastating diseases may finally meet their match in the form of healthy cells ready to restore function and hope to those who need it most.